Kid Next Door.
Hello everyone, welcome to the Kid Next Door. Today we shall be drawing a plant cell. So a plant cell we will draw as a rectangular shape. Plant cells have definite shape while animal cells don't have any definite shape. That's the main difference. So the first thing that we are drawing here is the cell wall, which is the outer boundary of the plant cell. This cell wall is made of structures like cellulose, and its function is to provide protection and support to the plant cell. Next to the cell wall, we'll draw a thin semi-permeable membrane called as cell membrane. Cell membrane is composed of phospholipids and fats. Its purpose is to keep a check on what's coming in and what's going out of the cell. Next, we'll draw a nucleus, and after placing the nucleus around its side, we'll draw a big, large central vacuole. So this gives like a basic outline to where we can place our structures. Inside the nucleus, there will be a nucleolus, right? And then next to the nucleus, we'll draw the interconnected network of membranes, which is the endoplasmic reticulum. So I have zoomed the view here for you to carefully see how I'm drawing these membranes, the thin folded membranes which are still interconnected, and part of it will be rough ER, the ones which has ribosomes on it, so it provides a rough view under microscope, and the remaining half without the ribosomes on the membrane is called as smooth ER. So in this way, we'll draw these thin folded endoplasmic reticulum membranes on both the sides of the nucleus. Okay. Next, we will draw Golgi apparatus, which are again thin folded membranes stacked over one another, and near them are also Golgi vesicles. So the proteins produced by endoplasmic reticulum, they come to Golgi apparatus in vesicles for storage and packaging. Now, on half of the endoplasmic reticulum, or ER, we'll put the ribosomes dots so as to give it a rough appearance, and this part having these ribosomes on it would be our rough ER, rough endoplasmic reticulum. The rest remaining one would be the smooth ER. Next, we'll draw the next important structure of the plant cell, which is chloroplast, which gives the green color to the plants. So it is a double membrane structure, and inside of it we will show the grana of thylakoid, like which is a stack of coins kind of a structure. So we'll just draw it with the random lines. You can draw as many as you want. So different plant cells have different amount of chloroplast, like leaves will have more than the stem. Next, we'll draw the powerhouse of the cell, which is mitochondria, which is like a simple sausage shaped structure. It is also double membrane from outside, and inside it has a thin folded membrane where cellular respiration occurs. So after drawing the double outline, we'll draw a folded membrane structure inside. So that's how the inside of mitochondria looks like. Again, you can draw as many as you have space in the box. Next, we'll draw a myeloplast, which stores starch in the plant cells. Inside the large central vacuole, we'll draw some crystals. These are different kinds of crystals, and the purpose is mainly again for storage. We'll draw the ribosomes throughout the cytoplasm as they are spreaded all over around and freely floating in the cytoplasm. In this way, our plant cell is complete, and now we can start to label it.